Welcome to the 9th episode of Junto Club. On this episode, we briefly discuss the inauguration before spending most of the episode on whether gene editing will deliver a utopian or dystopian future. For any questions, comments, or to talk mosquito extermination, email juntoclubpodcast@gmail.com. This is Junto Club.
Junto Club.
All right, welcome to the 9th episode of the Junto Club. This is a kind of group modeled off of Benjamin Franklin's original Junto Club, where he had people meet to discuss business, science, politics, philosophy, to try to learn more about the world and improve themselves. So we're trying to, vaguely resemble that, discuss those sort of topics along with what's going on in today's world. We're comprised of 3 PhD students from Georgia Tech. That's where we met each other. And, that's what this is.
All right. Well done. Welcome. So I'm thinking this way we can talk about, first of all, the inauguration, the big thing happened last week. And then we can move on to gene editing and all the ethical issues and stuff like that. Maybe we can touch on, ethical issue of robotics as well, because that's something that Mike is very well versed in, I think. So anyway, anything else you guys want to add to today's agenda, I guess?
Well, I'll just say the big thing last week was not the inauguration. It was Tom Brady making his 10th Super Bowl. But
What?
Okay. Notice sports was not in the list of things for this club, okay? Right. We're high-minded people here.
Fair enough.
Okay, let's talk about Tom Brady. What happened?
Well, okay, just briefly. He went to the Tampa Bay Buccaneers. It's his first year there. He previously had gone to 9 Super Bowls with the New England Patriots, won 6 of those. So this year, first year in the NFC, and he won the NFC, and now he's in the Super Bowl again with Tampa Bay.
And how old is he?
43.
Wow. Is he the oldest quarterback that's able to bring it to the Super Bowl?
Yeah. So he's breaking every record for oldest quarterback to win a championship game, throw a touchdown pass, all that type of stuff. Yes.
Is he still deflating footballs and cheating?
He's doing it better than ever, Matt. So
I think it's easier to deflate it in Tampa, Florida, because he's in Florida right now, right? So
maybe.
I mean.
All right. That's his secret, I guess.
Dude, yeah. I just want to ask him what sort of new age drugs he's on to stay youthful, because I want to get on those in another 10, 20 years.
We talked about this before. He published a book that actually talked about that.
Oh yeah?
Yeah. You know what he say?
What?
Drink more water.
Yeah, just like all the Avengers movie stars say, they just ate well and worked out hard, and that's how they gained 40 pounds in 9 months of muscle.
Right.
Yeah.
Yes, it had nothing to do with steroids, all right?
There were definitely no magical pills in their, professionally made meals.
Well, maybe Tom Brady has someone conducting some gene editing on him, very, very early stage, editing DNA in his muscle tissue.
I assume he's just being injected with stem cells directly into his heart or something.
You never know. I mean, they might have done something medical, right? Not medical, but something to making, never get old, I guess.
He's a robot. All right. Go back to your politics.
All right. Go back to inauguration. Actually, maybe Biden can get some lessons from Tom Brady so he can stay young as well, right?
You mean our president for probably 8 to 9 months.
Are you on the conspiracy that Kamala's going to have him offed or removed to take over?
No, he actually has seemed fine in what I've seen of him for the first whatever it's been, a week or so, I guess it's been 5 days, so he'll probably make it 4 years.
Yeah, and I guess that's on a lot of people's mind, I guess, right? It's like, oh, how long he's going to be president, right? So,
how many presidents have been replaced by the vice president? I mean, Kennedy, I assume William Henry Harrison, or whoever. Wasn't he the one who died of pneumonia like 6 weeks in?
oh, really?
Yeah. Yeah, he caught pneumonia. I think it was William Henry Harrison caught pneumonia at his inauguration and
That
died.
sucks.
Yeah.
Real sucks.
All right. So, I mean, all right. Let's not be a downer, Mike. You know, it's a celebration of America. We just, let us have some, happy days. Maybe at least 100 days. And then we can talk about the downsides. So, what did you guys talk about inauguration? Did you guys watch it?
A little bit of it. I don't really feel like much happened. It was pretty just, run of the mill. I mean, I guess it was different because of COVID and stuff and some extra security in case people try to riot again. But I mean, besides for that, I don't know. It was basically just standard, right? They did the ceremony. Biden started dropping executive orders and changes left and right.
Well, my policies, any policies that you think, any policies that you disagree or don't like?
I mean, I don't know. Are we getting $1,400 more or $2,000 more dollars? Do we know yet?
You are not getting, right? You're making too much.
Hey, I haven't filed taxes in 2020 yet, Shu, or for 2020.
Then you should return it.
Yeah.
Give it to me. I think that's the plan. So I think it should. I think the Congress, Senate are pretty much in agreement with of that. So.
No, so that'll be. I mean, that's the thing I keep seeing people post about is just the money. But I mean, student loan forbearance, student loan cancellation, I think is very much back burner at this point. But he did extend no interest, no payment through September, I think, of this year. So
Okay.
I don't know. And it seems like now he's actually going to get vaccines rolling out much faster already, starting to see increases in the number of vaccines per day. So I'm guessing these things might be the last extensions for COVID stuff before they're everyone's vaccinated, whatever.
Yeah. No, I mean, Amazon has also all of a sudden decided to volunteer to help distributing vaccines and stuff. So that's a big, powerful entity.
Not mentioning Starbucks.
Oh, Starbucks is. I didn't hear about Starbucks
Distributing the vaccine.
I think because this big company, especially in the Starbucks, they are very good with the logistics, right? Of getting things distributed across the U.S., right? So Starbucks is very good with that. And I don't think they are doing vaccinate people in Starbucks. That would be cool, That would be cool too, because they have very good customer service, right? So
that's right.
So everybody get coffee when you get vaccinated. Yeah.
No, there's better customer service.
Yeah.
Chick-fil-A. They could do drive-through COVID vaccines.
Chick-fil-A, the owner has come out and said COVID isn't real.
I mean, he's not going to miss a business opportunity, right?
No, that's fair.
No. Problem with Chick-fil-A is they close on Sundays, so.
No.
6 days a week, still a lot. And then who wouldn't buy a chicken sandwich while you're getting vaccinated? It's right there. Easy money.
No, I agree. I mean, every business, but I mean, I at this point, I feel the limiting factor really has to be the storage and I feel and the number of vaccines, right?
Yeah, I think mostly.
Yeah.
Yeah, I mean, we ran out of vaccines, right?
Yeah, I mean, I know there were a bunch wasted in the initial batch, and then places that were distributing them asked for more, and I think there weren't more. So, I mean, I think I don't know. Yeah. He wants.
Sure, they're been producing them as fast as possible for the last several months, so there will be more. I don't know how long, but
no. I mean, I think I don't know. Hopefully, if fall will be for Labor Day, things can be somewhat normal.
All right. So talking about I guess vaccine. Let's get into science. I guess so. It's time to talk about gene editing.
Okay, yes.
So, all right, Mike, take it away.
Well, so we were talking about CRISPR. So I tried to dig in to sort of like the mechanisms by which CRISPR worked a little bit. So I guess CRISPR, or sort of gene editing via CRISPR, is sort of based on what CRISPR's true purpose is, which is as essentially an immune, pseudo immune system for bacteria. So bacterial cells, relatively simple, but they can still be infected by viruses. And, the viruses want to replicate. So as a means of doing this, as a means of protection from being hijacked from viruses, they have basically, I guess, these interesting pieces of DNA that are they have repeated segments in that DNA. And then they have in between those repeated segments, they have what are referred to as spacers. And spacers, through various studies, they've found that these spacers match the viral DNA, pretty much exactly. So basically, there's proteins called Cas proteins. So, and that's CRISPR associated proteins, and Cas proteins are actually going to essentially when genetic material is inserted into the cell, these Cas proteins locate a very short sequence on the new genetic material, and will essentially unzip the cut the DNA there. And so there's sort of a 3-piece mechanism, the Cas protein, which is connected to this matching genetic material, via a different RNA sequence. Yeah.
Is that different between, I know the Cas9, right? It's Cas9.
Yes.
What does it stand for? Cas9. Do you know what it stands for?
CRISPR associated.
Oh, okay. So is anything different between enzyme and protein, or are they the same?
Well, I think
enzymes are a type of protein, right?
Oh, okay.
Yeah. Well, I think enzymes specifically carry out chemical reactions. So it's
Oh. So that's why it's not a scissor, right?
Right. So
you can carry out chemical reactions.
Right. So essentially, so in bacteria, an RNA is created that binds to the Cas9, and then a guide RNA or CRISPR RNA is created that binds to that, and when Cas9 unzips the DNA, at the location of what's called a PAM sequence, which is something. Oh God, I can't remember what it stands for, but it's something it's essentially a 3-letter sequence following what's included in the bacterial DNA, the CRISPR bacterial DNA. And so basically, when the virus comes in, PAM locates or Cas locates this PAM sequence. It unzips the DNA. And then the RNA, that's the guide RNA, will bind to it will bind to the virus if this is a virus that's been seen before. To essentially,
how do you know which location of DNA to zip, to cut?
Well, that's exactly so. Okay, so the Cas, so this protein will essentially unzip where there's 3-letter sequences, right?
Yeah, but how do you know there's so many some location of DNA right now? How do you know to go to the correct one?
Okay, so in the case of bacteria, the sequence that Cas9 actually identified is not included in the bacteria's like genome. Or it's not included in like the bacteria's the DNA in that bacterial cell. So it knows if it's seeing that 3 DNA sequence, it knows it's cutting a foreign DNA. And then essentially for the viral DNA to actually be like cut or or for it to be taken out, essentially, you need the guide RNA, so the complementary bases to match. So like that's a much longer sequence. So if they don't match, then essentially what you'd get is the cut DNA would be, or the unzipped DNA would allow it to be freely rotate, which in theory could allow it to like bind back together, right? So it's essentially like the binding that happens is very fast,
so they make a specific RNA that's able to find the correct, basically can go to the specific location that you desire.
Right. So in gene editing, so basically gene editing has a Cas9 protein. Usually, I mean, there's complexities that we're just not going to get into, but like there's a Cas9 protein. Yeah. It basically is bound to a single RNA molecule that takes the place of 2 different molecules in the genome of bacteria. And basically that guide RNA is just like the complementary bases to the DNA you want to alter. So basically Cas9 goes in, and the Cas9 you choose, so the protein you choose needs to be specially chosen because each type of protein, like it identifies like different short sequences. So you need to pick a protein that will identify the short sequence that immediately follows what you're trying to edit. And basically it'll identify that short sequence, unzip the DNA, then that like the rotating the half of the DNA helix will, if you're in the right place, will bind to the guide RNA. And that will cause the reaction to us, that will essentially like disable it or, do whatever, or like essentially replace the second half of the DNA helix that's no longer attached. So.
Okay. So the Cas9 molecule will basically potentially go through your DNA, unzip at multiple locations, wherever this 3 protein sequence is, or sorry, whatever 3-letter sequence, but only when it's at the right one will it's the RNA that's attached with it kind of bind to the DNA when it's unzipped. So everything else, it will kind of unzip and it'll just kind of rotate around and then end up rezipping on its own as it goes on, right? Is that correct?
That is my understanding, yes. So basically, that is why they talk about, so this, they basically say it's very important to get the guide RNA. So this complementary thing, it will work if it's not perfect. Like they say, it's it doesn't need to be perfectly aligned with the sequence you're shooting for, but it's got to be close. Otherwise it will not, I guess because there must be enough force to the rotation or something where it needs to be very close in order to fully bind.
So I'm imagining, so we know DNA is this double helix, right? So you unzip it, the things that were connected, rotate around, get bound to an RNA, but that's still all in your DNA, the longer DNA strand, it's just got this little section with an RNA binding to one half.
That is the picture that I've seen, yeah.
So that disables genes, right? But does that give you the ability to add a new gene that makes something, to make a new protein to do something?
You've only unzipped a little bit, right? So wouldn't it essentially be,
So I have some stupid questions right now because I think we're talking about protein, DNA, and gene stuff. I actually don't really know the difference between what's the gene and what's the DNA, and then protein, can you maybe give me a little bit quick, just like, what's the difference between gene and DNA?
So DNA is like the DNA is like kind of the whole thing or the concept, right? So you know how you've seen the double helix pictures in like biology books?
Mm-hmm.
So that's like all your chromosomes are your DNA. It's the whole thing. A gene is like a section of your DNA that does something specific. Is that right, Michael?
Yeah. So that's I guess. So you're replacing a single gene with this process, right?
Okay. So gene is a small section of your you can think of it as a tiny section of your double helix structure.
Yeah.
What's a chromosome?
So a chromosome.
Your DNA is in like clumps. So you have like each chromosome's like a literally physical clump that's of DNA. That's like a large structure. And we have like, we have 23. Humans have 23 chromosomes. So there's but the 23 chromosomes, they're not attached to each other in any way. You have each of these chromosomes in your cells. So you can think of them like each like a bundle of DNA independently, just wrapped up to get in their own little things.
Oh, okay. And they are inside a cell? Did you say that?
Yeah, in the cell nucleus.
And what about protein then?
I mean, proteins are just a very general category of molecule, and it's the main thing DNA makes. So when your body uses DNA to make proteins, so they are basically DNA is like the instructions on how to make proteins, and then the proteins do things in your body, like build cells and, almost everything else.
So DNA create these proteins, and then these proteins going to do whatever they do, basically.
Yeah.
Yes.
Okay. Nice. Thank you for the basic lesson.
Okay, so let's go back, Mike. You say it replaces that section, but you unzip a section of your DNA. You bind it to the complementary RNA, right?
Yeah.
That's not new, right? That's the same DNA RNA pairs you had before. So where's like a new gene go then?
Right. So, well, I mean, I guess you so this is essentially cutting, right? So essentially replacing. So I guess you would potentially provide new. So in order to edit it, you could provide new genetic material in the delivery package, right? I mean, I think that would be a potential way to carry that out.
What I think is that someone says that is like, so the enzyme, the Cas9 enzyme bind to the sequence and then basically deletes it, right? And then you basically replace it using some kind of benign virus that's engineered to deliver and insert the correct DNA, right? So you I guess the RNA and Cas9 is doing a deletion, right? And then you have another virus that is doing the insertion of whatever the gene you want to edit.
Right. Yeah. And I mean, also you can use Cas9, like you can use I guess a similar approach, right? To like once the Cas9 unzips and there's some type of deletion, like you could use the same protein to essentially like attach to essentially like fill in the gap, right?
Mm-hmm.
Yeah. So, I mean, so the Cas9 would then could essentially like identify the sequence that the previous protein did on the whatever side of the DNA is now free, and then attach the complement, right? So, or attach the genes you would want, or I guess yeah, you could just send in a vector that has the new genetic material, like the correct gene, and that because as I say, there's oh, that's right. What am I talking about? No. Like as I talked to, I guess, yeah, no. As I don't know what I'm talking about, but as I talked about last week, when DNA is cut, there's like essentially the body, or like there's a process of like automated repair. Like that's, I guess, she was asking about chromosomes, and you have 2 chromosomes from each parent. And I mean, I guess the question would be sort of why, right? And I mean, there's the answer in addition to like, I mean, I guess because I guess the way reproduction may have come about is because it's useful to have a pair, because when DNA is damaged, there's sort of a backup, right? Like, so even, I mean, this is a poor example, but like if, blood cell, right? Like if a blood cell's DNA is damaged, you have essentially the instructions from one parent damaged, but the instructions from the other parent like are still intact. So basically there's a process by which genetic material from one is used to repair the genetic material from the other. So when this, I guess, sort of unwinding or cutting happens from the Cas9 and the CRISPR RNA or the guide RNA, basically if you provide genetic material that's like a healthy replacement, I can't remember what the name of the process is called, but it was in the book, where essentially like they will uptake this like genetic material that's present and replace the gene with that healthy material. So.
What's the book you're referring to?
So the book is, I want to get the name right. So it is Cracking the Code, I believe.
A Cracking Creation?
A Cracking Creation. That's
Gene editing and the unthinkable power to control evolution.
Yes, that's the one. Yeah, and the authors, I believe, just won like the Nobel Prize in Chemistry. So, like in 2020. So.
Yeah, for her discovery of the Cas9. Yeah.
Right. Yeah. So, yeah, I mean, her work was on like the role RNA played in like immune response, I guess. So this was like, so she was excited to like, I guess another professor had approached her being like, have you ever heard of CRISPR? And she hadn't. But she like this professor talked about like how a very early on area of research was occurring that like pointed to like CRISPR RNA being like part of a bacteria's like immune response. So that's like what drew her to this area. And it sort of went from there. So
is it, so what did she talk about regarding the ethical issues of the gene editing? Like,
right. Well, I mean, there's always the question of like, I think in gene editing, there's a lot of the same questions that arise when people talk about like what could occur in robotics, right? Like, I mean, there are like exoskeletons already that provide like so there's like a discussion on restorative versus like enhancement uses, right? So restorative being someone loses a limb, right? Like a veteran, someone like that in war loses a limb, someone who's born without a limb. We can potentially provide an exoskeleton to replace that limb. And if we're able to attach it to nerves and they go through therapy, they could walk to what would be considered like a standard level.
You're talking about robotics.
Yes, in robotics. Yeah. Similarly, like with gene editing, right? Someone is born with some type of genetic disorder that, causes cognitive impairment or any number of other types of impairment. I mean, there's blood disorders. There's the potential for like genes, like people can be born with mutations in genes that predispose them to cancer or Alzheimer's disease. Like they've identified genes and gene mutations that skew like the likelihood of people having these different things, right? So the idea would be like, so to restore is to essentially fix the mutation. So do what we've sort of been trying to talk about poorly, which is basically just like, delete the mutation and replace it with something that creates the proteins in a way that aren't going to predispose you to these things. or, delete it and fix the mutation that is causing these different issues. That's creating like essentially an incorrect protein, or creating something that's harmful to you, or not creating something that you need. So that's sort of restorative, like replacing the missing limb, or correcting a mutation that we found to be potentially hazardous.
You can replace a missing limb with gene editing?
Well, no. I'm saying robotics. So I'm saying, because that's what we're used to discussing. So those would be like restorative purposes, right? So like you fix the mutation. Gene
editing and robotic just, I guess, confused me a little.
Okay. So fine. You fix the gene mutation that has been shown to be harmful. So that's restorative. I mean, enhancement is you could make, particularly if like someone is like in utero, like it's a baby who's growing and like at the very beginnings of their existence, you could alter the DNA to make them, like, we could identify genes associated with like intelligence. We could identify genes associated with like health and aging. And we could alter those genes or we could identify genes even like associated with beauty, right? So like we want this baby to have like super metabolism, so it's skinny no matter what it eats. Have blue eyes, brown hair, or blonde hair, or whatever. And
Mike just says his own eye color and hair for beauty.
Yeah.
Why did you mention blue eyes? What's wrong with brown eyes?
Because Mike has blue eyes, and he's proud.
Well, I'm just okay. If you want a baby with brown eyes, that's the thing, right?
Also, why did you first came to like when you first say, design a baby? Why did you come right away? You say blonde hair, blue eyes, white skin. What?
I didn't say anything.
Why did you came to that first?
All right. Well, the first thing I think I said was intelligence, and the second thing was health. But yes, no. I mean, I guess because literally this ethical question is often described as creating designer babies. So it's what the design industry might push if it.
So what's the ethical problem here?
Yeah.
Basically, often what it comes down to is disparity between wealthy and non-wealthy, like most things. So basically, wealthy people can have stronger, smarter, like quote unquote better babies, and and make themselves healthier. I mean, obviously, if you're editing the genome of editing your genome, you can maybe fix problems that other people couldn't fix, or give yourself give yourself an edge over people who can't afford these type of treatments.
Yeah, but isn't it the case already? I mean, rich people have more medical care, right? So they live longer, live better lives. So this is the case already, right?
Better quality food, their kids get private tutors, better schools. I'm concerned here that we're all going to be in basically agreement that, I'm aware that it's a concern some people have, but it's not real. I don't know. Is it a real issue? Rich people will get it first. And like all other technology, it will spread out until, the masses can use it as it gets cheaper and more available. And then, everyone's healthier and better.
No, I mean, I basically agree. I mean, but it's once again I again go back to robotics. So in robotics, we have like these restorative things that, like limbs that essentially, like, if your limb is missing, we can replace it with a different limb, and you can walk and live sort of quote unquote a normal life. But okay, so what now if we have like an exoskeleton that gives you super strength? Like, should we restrict? So essentially the debate we have in one of the classes I TA at Georgia Tech, like one of the debates was like, should we have an exoskeleton and restrict it to where you can only lift what's typical for people, your age and, whatever. So, as opposed to like basically, hey, your family is worth 100 million. Like you can just, throw cars at people if you're upset. And, you know, if you're poor, like you can't afford an exoskeleton, so you can just hit the gym more.
So how did you think about that in your ethics, robot ethics part? How did you guys talk about it? How did you guys think about it? Did you come up with a resolution from the answer?
Well, I mean, that's the thing. I don't know that there is like a simple, so I guess it's depending on the ethical framework you want to use and like how you do the analysis to come to that conclusion, right? I mean, because if it's like more like deontology, then you may lean in the direction that there should be some types of restrictions here, or like like you need a.
What are the ethical frameworks and what is deontology?
Well, okay.
So
So,
basic ones, I guess.
well, yeah. Well, I mean, it's like, I mean, the big two, or sort of like the two we used like most consistently, were like utilitarianism, which is basically like you consider all stakeholders like in a problem. So like all the people that the decision is going to affect. And then you say, like, basically you find some way of quantifying how, like, so there's some number put to like the utility of the decision. So whether it like, so for each stakeholder. So it's like if you were going to put out a product as a company, your stakeholders would be the customers, those or those affected by the use of the product, the board of the company, the employees of the company, all those different people. And say like the product, say, you know, it's something like junk food, right? Or something like that. Mm-hmm. So the net, so there may be like a net negative for the user, right? Because it's like very brief enjoyment and like, but like the net negative is
utilitarian basically is greater good, right? If it was,
it's greater good. Yes, it's greater good. Yeah, exactly. So basically, you make the decision in one way, the way where it's a positive value, and you don't make it in the decision that's a negative.
For a certain majority of people,
right.
Yeah.
Right. So basically, it's a weighted sum. If the sum is positive, you do it. If it's negative, you don't. Gotcha. Deontology is more about considering considering people as individuals. And sort of the guiding principle of deontology is like people should never be used as a means to an end. People should be essentially treated as ends in themselves.
Okay.
So for example, like deontology would say you shouldn't lie, because if everyone lied, then like that would be a bad thing. So basically, like you shouldn't lie to people because you know not everyone should lie, and therefore you know you're making different rules for different peoples essentially. So
is deontology similar to virtue ethics? Like you should be good for good sake. It's not because you should be good because the consequence is good.
Maybe. I'm not. I guess maybe I don't know enough about virtue ethics to like clearly differentiate them, but it sounds similar. So in any case, like often these discussions are like there isn't necessarily like a clear like this is the right thing to do, right? It's more just. I mean, again, it's sort of like the types of discussions you have in politics. It's like people can argue, oh well, if you raise taxes on the top 1%, like we're going to be able to do like afford more services for the majority of people, right? But or like what we were talking about last time, if we allow science and technology to advance, it's going to be beneficial for the like in the long term for the most number of people. But there are people who are losing their jobs with innovation who are going to argue against it because they're like, you're hurting this group of people. And the 1% is going to say like, oh, well, you know, we're not going to have the money to invest in innovation and, you know, trickle down type argument. Right. So, I mean, I think
so you're basically saying there's no really clear answers to these kind of questions.
Sure. I mean, I won't say like, I mean, maybe as time passes, the answer becomes clearer and clearer. For example, like if we're like, oh, we're going to go for it, like essentially if we take the route we may largely agree on, which is like, okay, like this technology is coming, this technology is going to be hugely beneficial for society, like let it just go with essentially no restrictions. And then 100 years from now, there's essentially superhumans and essentially like humans are a secondary class of people. And then there's like a couple thousand superhumans that are essentially their gods. Then maybe humanity would look at that future scenario and say, well, that's no good, right? Because like there are billions of essentially like people who are now secondary class citizens and only a couple of thousand who are gods. But if we look forward and we see like, oh, like class disparity is no worse than it is now, and basically we don't have things like, Alzheimer's is far, far, less of a risk, and people are living to be 150 now on average. I mean, I think everyone would sort of say, like, yeah, we should go for it.
So, so besides, you talk about the one issue, designer babies, right? There's a, ethical issue regarding that. What other ethical issues are there besides that?
Well, I mean, I think what it all boils down to is just, of course, I don't even necessarily consider it as much, but sort of like the quasi-religious or pseudo-religious one, like, should we be playing God? As we change this, like, what types of questions does that raise about our humanity? Things like that.
I was going to say, with babies, the questions become a little more, I would say, reasonable. Like, because if you're just gene editing yourself, I feel like you got a big hill to climb to say that's wrong, right? But there are people who are like, I mean, obviously parents have a huge amount of influence and control over their kids, how they grow up in their lives. But there are people who, say there should be limits, right? You're not supposed to, you're not supposed to do certain things to children. It's still their body. You should let them grow up and make these decisions for themselves. I've heard that with regards to like piercings or circumcision or other stuff like that. So, I mean, I could see people making the argument that gene editing your child, or, even if it's like at the embryo level, right? Like it's like kind of like before they exist, like you make them, but I don't know. I could see people making an argument that's like some sort of improper control, but the flip side's really just like random, right? It's like you're controlling something rather than it just being arbitrarily random, which, when you look at what the other side is, it's like this, there doesn't seem to be a clear like route to this being bad.
Yeah, I think the main concern I have heard is something called gene drive. So basically you design, for example, you design your baby, right? You have a gene. Mm-hmm. That means the problem with designing that, you basically, the gene going to be, whatever you edit, it can pass down to generations, right? So you don't know what's the consequence of that, right? So if you design something and then you just because it can potentially propagate through the future generations, through many generations, through spread to many other people, right? And then the danger of that is if whatever you maybe something you change, you thought it was good idea, but you don't know the consequence of that, and the consequence of that consequence, and then the other consequence of that consequence, right? But once you plug it down, then you don't know what's the effect on the whole human race, right? You're basically changing the human race in a way, right? Because the DNA propagates, right?
Yeah, but you could also have a recessive gene for some disease that you spread to your child, and it spreads, and that's how a lot of the diseases get passed around that exist, right? You say it's not like this is something you figure out in one generation, right? Like if a gene causes a problem, unless it's recessive, I guess. I assume if you're editing it, it's either going to be dominant or you'd have both genes be edited, right? So to make the impact known. So going with that assumption, like you would know the next generation, like you'd see what the impact of the gene was and know whether like, oh shit, like something bad has happened that we didn't realize or not. And since this isn't like a universal thing, like you'd have some people using it, we'd figure out the long-term effects over their lifetime, and then we'd kind of know. And then if it turns out good, probably become more common. If there's an issue, people would stop doing it.
Mm.
But like I said, a big part of this is like you're controlling versus random, but like you could always have some random gene that fucks you up too, right? So, and you don't know if you have a kid, they might get, unfortunate genes that cause all sorts of problems. I mean, that happens every day, right?
Right. I mean, there are mutations
might happen
now.
every day.
There are people born every day with like serious genetic diseases, and that impacts their life tremendously and or just kills them. So.
Right.
Yeah, but these people, if they have disease, they won't pass it down, right? Because sometimes they usually die early, right?
So sometimes depends on the disease.
Mm.
Mm-hmm.
But I mean the same thing, right? Like if it's a very serious, like if you use some new gene, right? Like some gene edit thing that causes a major problem. I mean, you're right. They're probably, if they're really seriously impacted from it, they're not probably won't have kids in the first place. They might be super sickly. They might just die. They'll probably at least be aware that it's an issue and maybe not just try to have biological kids. Like, I don't see how you have a DNA that somehow like doesn't do anything for several generations and then like suddenly like pops out and it's like, surprise, everyone who has this gene just got activated.
Mm.
All right. Well, sorry. I was just because I wanted to be sure we were answering Matt's question with more than speculation.
Okay.
So we can go back to the ethical discussion in a minute. But I was curious. So CRISPR cleaves the double-stranded DNA.
And you say cleave. Do you mean like a cross or like you mean split them, right?
Right. So it splits the DNA. So it splits the DNA and leaves that gene damaged, right?
Okay.
So basically repair can happen in one of two ways. So enzymes will essentially stitch like the dangling ends of the DNA back together. Where the break took place. Or it will which can result in dysfunction, some type of dysfunction on its own. Or let's say so. Alternatively, the other enzymes can patch the break with a single strand of DNA that matches the DNA sequence upstream and downstream of the cut. And I guess a complementary DNA strand that is created to complete the double strand. So like where so essentially like if the gene is missing here, like you can essentially like just try to meld it together, or you can like create something to complete that and something to complete that. And I guess that in the case, so if you provide genetic material with like your cutting package, then that can successfully create or successfully like replace the gene that was sort of taken out. So, but I mean, this is like an article from Science Daily from like 2019, and it's basically saying like the science behind like repairing, like getting the right repair and not sort of messing things up is still, I mean, obviously we don't have like
still not mature yet.
Right, exactly. It's still not a mature science yet, so that's why. Like, so there's still a lot of questions about how to like this is CRISPR is revolutionary in how precise it can be in the deletion, but in actually getting like the repair part right, it's still not fully matured.
Mm.
So, yeah.
We just gotta. Has anyone thought about just making a new chromosome and just sticking it in? Think about that. Nobel Prize, here I come.
Oh, that's all right.
You should get another PhD in chemistry.
Next on my list.
Yeah. That's 40-year-old Matthew O'Brien in his postdoc. So, yeah, no. So, I mean, basically, I guess we still have a few more years before this is it's similar to robotics, which has all these discussions, but as they say, it's now time to have these discussions before the technology is just here. So
going back to Matt's idea of chromosome, I don't get it. Why is it significant?
Well, the difference with that is rather than trying to insert the gene into the middle of the DNA strand of another chromosome, you just kind of have a new chromosome floating around that you made. So you don't have to put it into, like, you don't have to mix it with other DNA, right? If you could get that into the cell nucleus, and I don't know of any reason, like, I mean, I assume once you have a chromosome there, your body just treats it like all the other chromosomes, right? So I assume that way you could like make the DNA you want and then just have it floating around in the cell nucleus, making proteins and whatnot. But again, I'm not a biologist. We really should get one on this show if we're going to talk about stuff like this.
That is fair. But I guess also that, like, I mean, essentially that takes the parent out of it, though, right? So, like, I mean, I guess depending on, so if it's the DNA, like, specific to, like, a certain, like, area of the body, I guess that would be good. But I mean, I guess if this is a baby who is like relatively early in development, and you essentially are like inserting a chromosome, like you immediately see like, oh, this, the chromosome has this mutation in it, like that would essentially, and you just stick in a chromosome. I mean, I guess it would only be essentially 1/23 of the parent that's coming out, but.
Well, I'm not saying you replace the old chromosome, right? Just a 24th one, but one that's designed to do specific things, right? So, like, what I guess the thought was, CRISPR is good at deleting genes, right?
Right.
So you're given an embryo, 23 chromosomes, lots of DNA, and you say, oh, look, these like 19 genes are all fucked up and going to cause diseases and problems, limitations, whatever. So you send in CRISPR, delete them all. You don't bother adding anything in because it's hard to insert the right DNA in the right spot and all that stuff. Then you just have your 24th chromosome, like a software patch that just put it in. And every cell will now have this 24th chromosome that has just the genes you need. So it's got the replacement for the ones you deleted, and then maybe a few other genes that give you some nice things like, oh, these two genes really make you almost immune to cancer because, blah, blah, blah. It causes cancer cells to kill themselves way better.
Right.
Add that patch in, now we are 24 chromosome beings and, whatever.
Don't they?
Well,
I just heard something that remind me, what you say just remind me of something I heard recently. It's like, I think there's a baby somewhere and it was born with like 3 people's DNA. I think one lady and then there's 2 guys' DNA. I don't know how they did it. I think that something was missing from the parents and then they just get something from the other person.
Well, that's just like the ribosome DNA or whatever. I think
the mitochondria.
Oh, mitochondrial DNA. Yeah.
Yeah. Is it similar to your idea, Matt, or no?
Not in the specifics, but in the broad sense. Well, mitochondria. Do you know what mitochondria are?
I know it's not really. Go ahead.
Mitochondria are little organelles of the cell, like an organ of the cell that kind of makes the energy, ATP. But they have their own DNA. So, so it's kind of like an extra. So you get, a chromosome from each parent. So that's normally why you have you have 2 people's DNA. Yeah. But normally you get the mitochondria of your mom, but they replace it. Now you have the mitochondria and its DNA from another person. That's kind of like a third person's DNA. But I mean, it's kind of similar in the sense that here's like a clump of DNA that can do something, but it's not part of the other DNA that's doing other things. It's just, separate.
Okay.
Yeah. I mean, go ahead, Shu. Sorry.
I was going to talk about some CRISPR babies in China. So, I mean, I think before we move there, I think you can say whatever you want to say.
Well, I was just going to ask Matt, because we know Down syndrome is caused by a partial or full extra copy of like the 21st chromosome. So I'm just wondering if.
Well, yeah, but that's an accidental thing, right? Similarly, we already know, like, if you delete an important, like if an important gene gets deleted or mutated away on accident, right, it can cause a disease.
Mm-hmm.
But CRISPR can also fix a disease by deleting a specific gene intentionally, right?
Right.
So, I mean, as far as I know, there's no reason fundamentally that an extra chromosome would break the human process. Again, not a biologist. But it makes sense that if you have random DNA extra there, I assume the issues are related to extra proteins being produced.
Right.
Obviously, the chromosome's being used if it causes any changes. So I assume it's meaning more of this stuff's being produced, and that unbalances something and causes other issues downstream. I probably should be Googling this because I'm really just speculating here. But I feel like it's pretty clear that if you did it intentionally with the right genes, it probably wouldn't cause problems.
Right. Yeah. No, I mean, that does make sense.
I just want to say that I think speculation is the first step to science.
That's true. We just got to be clear about what we actually have expertise in and what we don't.
Right. No, I mean, I have no expertise in this, but I find it very, like, I mean, I want to learn more because now I'm wondering, because I know very little about going from DNA to RNA. So I wonder if like the transcription process like would require some type of ordering, or like, I want to know what the script that the body uses to convert, like if the script is overly hard-coded or not. Because like if the positioning of the genes makes a difference or not. I mean,
like, maybe, because maybe like you want some genes to be like all ran together, right? So you put them in place, and every time they're copied up all at once, and that kind of keeps certain things balanced. I could see something like that being a thing. But, it's a lot of speculation. A lot of these questions might not be known, period, right? Like, obviously, humanity in general has not mastered gene editing, but...
Right. But no, I mean, it's... Oh, go ahead, sure.
I was going to say, that reminds me of another ethical issue is people could use it for, like, as a bio weapon, right? Instead of, like, could be in the future of atomic weapon, basically. And that's one of the mass. Actually, I think recently, in the book, she actually mentioned that gene editing, CRISPR, had been classified as one of the weapons of mass destruction, right? Potential in the future, right? Like one of the four, right? One, the nuclear weapon. The other one of them actually is CRISPR, right?
I guess I guess I'm trying to think. I mean, I know CRISPR. I mean, where this has made a big difference already, of course, is like agriculture, right?
Yeah.
Like, because it's like you're making healthier food. Like GMOs, like they're bad. They're bad. So I mean, healthier crops, like crops able to grow in different environments. I mean, so maybe. Like, I'm trying to figure out how they're thinking of it being used as a weapon. Like, maybe modifying.
No, you can, if you know, you can modify something. I'm not sure. Somehow you can target it. You can target a whole population of people, right? With something specific, engineer something, virus, I guess. I don't know. Like, target something. Right?
Well, I suppose that's true. Yeah.
Weapon, right?
Right. No, that's true. Make us essentially. Well, that I guess must be it. So it's just a virus. Like make a virus that's.
I know one of the reasons because so gene editing, like cloning, right, is very hard to do because it's so expensive. So that's why, even though we all know cloning human is illegal, and there's no like mad scientists try to do it, right, because it's super expensive, right? Maybe someone's doing it right now. But still.
China's definitely not trying to clone people, wink, wink.
Possibly. So one reason about CRISPR is so cheap and very easy to do, actually. This is one of the major issues, right? You can actually, like, probably you and us probably can just buy a kit, like, for like 100, like, cost like 100, like 20, less than $100. Right. You can start doing actually CRISPR gene editing yourself.
Yeah, I was actually looking at it. I'm like, oh, this might be something I want to do in my spare time.
Okay, hold on. So you could have, but like, how would you make, like, even if you knew the gene you wanted to, let's say, delete,
right?
Like, how would you make, like, if I was like, oh, I really want to delete this gene, how would I make the complementary RNA piece that I would put into the CRISPR to, like, how would you make the RNA?
I had no idea, but I'm just saying the tool is right there. It's only
I'm not sure it's quite
$100.
that easy, but I get your general point.
The technology is very easy to use. It's only less than $100, right? Yeah.
Yeah, no, I mean, it may, like, there may be, like, as you say, there are kits and there may even be places that have, like, synthesized, like, so, I mean, obviously it requires probably, like, a ton of money and time to, like, understand each thing, but I guess once they do, like, the synthesis process must be pretty easy, so.
Yeah.
Look, all I want to say is, I'm not saying it's cheap to clone a human, but we've cloned enough other mammals that are basically no more complicated or, you know, different than humans on, like, on a fundamental, like, what makes something hard to clone or not level. There's probably already cloned humans, I'm just saying. I'd be surprised if no government has ever done it and just, like, either killed them or just kind of, like, let them go live at an orphanage and not tell anyone.
Yeah. Well, yeah. So I guess I can see that conspiracy theory going somewhere, but yeah. The main point is it's more expensive. Cloning is more expensive. Not like regular people can buy something for less than $100 and clone yourself, right?
Yeah.
So,
yeah. No, it'll.
Well, also, since you mentioned in China, I guess we need to talk about the China CRISPR baby. Have you? I think a few years ago you heard about the Chinese scientists like edited the embryo, right, and it resulted in like 3 babies.
But were they actually brought to, like, were they actually living, or did they just edit it in?
Yeah, those 3 people are actually alive right now. Like a couple of years ago, like I think 3-4 years ago, maybe around 2017, right? These Chinese scientists, basically, the parents has HIV, right? So these parents want to have children, right? So he basically, what he did, he edited in the gene such that the baby are immune to HIV, to AIDS. So so those actually baby were born, right? And then actually the scientist is, I mean, of course, when the scientists publish the results, right, the data and the result, whatever, and conference, you know, it get like condemnation from all over the world, like people just like freak out, right? So, and also after that, he actually got sentenced to 3 years in prison in China. So.
Yeah, prison doing work, lab work for the Chinese government.
Yeah.
They'd be like, here's your prison. It's the, you know, world-class biomedical facility.
Right, exactly. You can just never leave.
Yeah. Well, okay, so another issue I guess I want to talk about is mosquitoes. Application of gene editing. Have you guys heard of that?
Oh, is there some way we can kill all mosquitoes with gene editing?
Yes.
Ooh,
so basically, what you do is you basically engineer something. It's called you cause one mosquito to be infertile, right? So they cannot have babies, right? So once that gene got passed down through the whole population, you kill on a genocide. What do you call that? Cause them to go extinct, right? So that's the idea.
Yeah, I have heard of that. They make these mosquitoes that they can. It's important that they can have children, and they're apparently very like sexually attractive. You know, hot mosquitoes or whatever. So they'll mate a lot. They'll have a lot of kids, but their kids are sterile. So then, if they mate a lot, then they'll produce like a big portion of the population will now have sterile children. So suddenly, like a large portion of like the mating doesn't produce any offspring. I know it's. I think it hasn't it already been used to like cut down on population, but I don't think it would make them extinct. Just because you're never going to get like every mosquito. You're never going to stop every natural mosquito from like having babies, eggs, whatever.
Yeah, well, I think they try it a little bit in in South America for the Zika virus when it was really crazy. And I think that's one of the issues that people want to talk about. Like, do you really want to get rid of the mosquito? Because if you remove all mosquitoes, you don't know what's the consequences, right? Even if something like mosquito, something that people will agree, yeah, we should get rid of, but we don't know the consequence of that.
So I've seen stuff that suggests scientists believe the mosquito is actually not an important part of the ecosystem, and you could remove them and it would be okay. And I'm going to trust what I've read because fuck mosquitoes.
Consequences, Matt and I don't need to go and buy tiki torches and get dirty looks, Shu. That's the consequence.
Well, what's tiki torch?
I just took this in a whole different direction.
No.
My.
So, no, but I mean, I think it would work if you put them in if you put the attractive mosquitoes in enough areas.
I mean, it would definitely cut down on population, but how do you ever get them all with that? You know, like there's going to be like
simultaneously like cut resources, like hit them on both fronts, right? So like have these like super breeder mosquitoes that like make the next generation, like make that the baby boomer generation of mosquitoes, make it like huge. And then make the resources as limited as possible for them.
The resources is like anything with blood. Like, what are you going to do?
Well,
wipe out all native animals to get rid of the mosquito population?
Well,
I guess even if you did this, let's say in Georgia, you know, we all were at Georgia Tech. Georgia South has a decent amount of mosquitoes. You know, if there's some place in Texas, some random ass place that they didn't get them all, they're eventually going to spread again, right? Oy. It's just, there's no way you're going to wipe them all out with this. Because insects are all about, like a few insects can make thousands of eggs, right? It's a very robust biological species. It's just going to be very difficult.
Okay, mentioning Georgia Tech, I just want to say Georgia Tech does not endorse this podcast.
Yes.
Fair enough.
These ideas are our own, and no one endorses any of them.
Yeah, anything we say controversial is not. Georgia Tech does not approve.
All right. Well, I think we've run the mill on gene editing. Was there anything else we wanted to get to today?
I guess. Well, what's our resolution on that? Are we deciding that utilitarianism would say full speed ahead?
So next one, I need to go back to Richard Feynman, right? So you guys know Richard Feynman, right?
The physicist, yeah.
Yeah. So I consider him almost like the modern day. Well, I guess he's dead now, but almost like the modern 20th century Benjamin Franklin, because besides physics, he does art. You know, he studied a lot, and then he also talk about he very good communicator, right? Teach science to make space in the population. So I got a quote from his book actually. So this is a book this is a quote that he actually got it from a Chinese proverb. So he says like. And the quote says, quote, to every man is given a key to the gates of heaven. The same key opens the gates of hell. Right? So I think he kind of talked about this in almost a little bit. We kind of talk, I think because he worked on the Manhattan Project, right? Atomic bombs, actually. So I think he kind of think about that in that context as well. And how, like, science, you discover something, right? But this is not a key, right? And how you use the key is like which door you want to open with the key, right? So the problem is that we don't know which door we open, right? Even though the key, the technology is, the science, the key is very useful, has values, but how do you use it? Now what's uncertain is which door to open, right? To heaven or to hell, right? So anyway, so even people like him, I mean, doesn't really have the right answer to these kind of issues anyway, even to atomic weapons, right? So, yeah, so basically, anyway, so that's something that to keep in mind, I guess, that I'd like to share. Any thoughts on that? On the quote?
Man, But I mean, it's true that, I mean, I sometimes think about like, I mean, so every man is given the gates to heaven. They also open the gates to hell. Like, I guess, so there can sort of be like so ignorance is bliss is another saying. And I often think about like, that's coming from a place of like intelligence and like an understanding of like the world around you can sometimes, like, obviously it's beneficial because it enables you to do it enables you to do a lot. It enables you to better interact with people. But it also can be difficult in the sense of like, you may not want to know certain things. Like you may because I brought this up to you sort of jokingly, Shu, when we were talking about like last week when we just talked on the phone when Matt's internet didn't work. Mm-hmm. Like I was like, oh, the more I learn about like the underlying, like how humans work, because like as Matt said, like CRISPR very much could function like a software patch, and I'm like, are we living in a simulation show? So, I mean, I guess I guess there's a certain depth that is beneficial, like to understand how things work it's beneficial to understand like how to eat healthy, right? Because you live a longer, healthier life and can do more. But like at another level, like understanding health and sometimes recognizing the random like perturbations in health that can be detrimental can also be like anxiety inducing. So I guess this is a very long and like poor path to be like, every man is the keys to open heaven or hell, or every man has the keys to open heaven and they also open the gates of hell is sort of like, I view it as like, well, every man has the potential to learn and to like be like grow as a person, but at a certain level, like that can, like you can in a way go over a cliff, right? And become like obsessive, or like if you get bogged down in the complexities of the world, universe, whatever, like it can send you over a cliff. The more you do it.
That's why you need to keep it simple.
That's right.
I feel like this quote is just a more elegant version of guns don't kill people, people kill people.
Whoa.
Same thing, right?
That's pretty deep. Wow. Are you a gun nut?
I mean, I'm not on the own a gun, so I don't think I'd qualify. But Shu knows that me and Mike are generally pro Second Amendment.
Yeah, actually, yeah. And then that's a good. I think maybe you should use this quote. What do you call that? Call an RNA. Say, hey, you should use this quote as a excuse to have guns, right?
Did you call the NRA the RNA?
Oh, it's mRNA?
Getting things switched up here.
Junto Club.